Hello guys! So welcome sa post-laboratory discussion natin on our bacterial transformation. Okay? So the plasmid that we use is pGLO. Small letter P, big G, L-O, pGLO. So anong gagawin? It will light up your E. coli. Okay? It will transform your E. coli into a glowing one under ultraviolet race. So, simulan natin. So, why genetically modify organisms? So, ano bang reason dito sa genetically modified organisms? So, basically, their genes were engineered. Kung baga nga, it's modified. Yan. So, para saan? Modified mosquitoes to fight disease. Yan. Paano kaya to? I want you to search this, okay? So, paano na GMO ang mga mosquito? RBT corn that is pest resistant, diba? And also, they could fortify, give the corn different food, fortified nutrients. Yan. What else? Modified animal research models, pwedeng mga hindi nagkakasakit, na rats, na rabbits, and production of insulin, hormones, vaccines, and anti-cancer drugs. So guys, genetically modified, genetic engineering, DNA recombinant technology has really changed medicine. And actually, pasimula pa lang to, okay? Post-lab discussion pa lang to. So back then, guys, so insulin was produced from a bovine, okay? Or ano ba to? Cow. Yan. So dati, ang insulin kinukuha lang sa mga bovine. So actually, that was not that effective. Pero in 1922, it has changed the world because at least naging curable na ang diabetes, okay? Especially type 1 diabetes. In 1978, scientists at Genentech, yan, so produce human insulin using genetically engineered E. coli. Yan. So doon nagsimula yung recombinant DNA. So they changed the gene of the E. coli through its plasmid, okay? And 1982, Humulin was approved by FDA. So kung di ako nagkakamali, guys, itong Genentech, they were the one who made Humulin. So Humulin is the genetically modified insulin. Yan. So I will discuss in how insulin was made in through your E. coli. Pero the same thing, they inserted a plasmid to your E. coli. And what they did, guys, is they made mini factories. Ginawa nilang mini factories ang E. coli. So, malaman natin paano nangyari yon. And you know, guys, sobrang amazing itong biotech industry. Ang gold, sabi niya, $40 per gram. Hindi ko alam kung anong year to, anong basis na to. Ang insulin for diabetes, it's sold from DNA recombinant technology, $60 per gram. Human growth hormone for growth disorders, 227,000 per gram. And granulocyte colony stimulating factor. So, hindi ako nakakamali. Ito yung pang mga leukemia, kung leukemic patients, yan, cancer patients, it's 1,357,000 per gram. Yan. So, this is a big industry. Talagang nag-uunahan itong mga biotechnology companies para mauna sa next na biotechnology product. Grabe, no? Maglaki siyang industry. So, gone are the days na talagang gold ang number 1, oil ang number 1. So, marami na talagang pagkakakitaan. So, how could we make lots of protein? Yan. So, kung babalikan natin, huwag nyo kakalimutan yung concept that proteins are coded, diba, translated from your DNA. Diba? Lahat yan nagsisimula sa DNA. You need a gene. Yan. That will code for that protein. So you need a gene. In this gene, will be transcripted into RNA or mRNA. Tapos itong RNA matatranslate sa protina. Okay? So that you could make lots of protein, you need number one, that specific gene that will code for that protein. Yan. So kailangan hanapin muna ng mga scientists. Yan. So we could cut it. Yan. Itong mga gene na to, hindi by using your restriction enzymes. And you could put the gene into the bacteria. Okay? So dito, mukhang na-edit na. Kunyari, this is your plasmid already. They inserted already the gene that they want to be created. And they inserted to the E. coli. Okay? So guys, ang key naman dito, hindi lahat ng E. coli could uptake, kaya nilang i-absorb yung plasmid. Okay? So that's why kailangan i-isolate. Kailangan i-incubate yung E. coli. So, mamaya, mapapakita ko sa inyo, okay? So, ang gusto kong ipunto na hindi lahat ng bacteria. So, once you expose them to the plasmid or to that gene, hindi lahat ma-absorb yung plasmid, okay? So, kunyari, kung skincare, ganyan. So, may mga tao lang talaga na hindi hiyang, kumbaga dun sa skincare, hindi pumasok yung skincare sa skin nila. So, parang E. coli rin, okay? And so what they do is put the gene into the bacteria and grow lots of bacteria. So to multiply that gene, okay, they should multiply the bacteria through binary fission, di ba? So ang galing nila, guys, big brain. So pinasok nila yung plasmid sa loob ng E. coli, and now this E. coli replicate, or they multiply through binary fission. Hindi ba yung isa nagiging dalawa, yung dalawa nagiging dalawa rin, tapos yung isa naman nagiging dalawa rin, and so on. So it's multiplying very fast. Hindi ba after 24 hours lang nag-culture ka, nag-grow na sa culture media itong E. coli, as long as you give them the proper nutrients, patuloy lang siyang magmumultiply. So, kasama ng pagmumultiply, guys, ng E. coli, sa loob niya is that plasmid, di ba? It's that genetically engineered plasmid. So, kasama ng bacteria na nagmumultiply, yung plasmid mo rin nagmumultiply, okay? And the bacteria transcribe and translate the gene being mini protein factory. So imagine if you have so much colonies of E. coli, and that E. coli has that gene, tinranscribe niya, tinranslate niya, at nagpo-produce siya ng green fluorescent protein. So the production of green fluorescent protein ang pag-aaralan natin ngayong araw. So kunyari, kung yung E. coli na genetically engineer mo nga, nilagyan mo ng pGLO plasmid, tapos sabihin natin, kinulture mo for 24 hours, so it has multiplied, at saka yung pGLO plasmid mo rin, nag-multiply rin siya, okay? So, nilipat mo sila sa ibang culture na naman with the fresh nutrients. So, after 24 hours na naman, nag-multiply na naman yung bacteria mo, yung pGLO mo, nag-multiply rin yung proteins. So, they serve as mini protein factories. Naintindihan ba? Okay? Out of that gene that you have placed in your E. coli, it has divided so much and it has produced itong mga E. coli na sobrang rami na, nag-produce na sila ng protein. Yan. So imagine kung insulin yung in-insert mo sa E. coli. Ang rami ng E. coli na may insulin gene, right? And now, yan, they could now produce insulin as a protein. Okay? At pwede mo na i-harvest. Yan. So, dapat hindi siya masyadong mahal kasi ang dali lang niya i-produce, di ba? Mas produce. So, kaya nga, di ba, yung vaccine natin, guys, ang bilis nila rin i-mass produce. Yan. So, ito yung protein. Yan, ang raming na-produce na protein kasi marami yung bacteria, mabilis rin siya mag-multiply. And what they're going to do is purify the protein. So since may iba pang protein, siyempre naman ang raming genes maraming genes na nagko-code sa different proteins. Siyempre, ang ikukunin mo lang na protein, yung protein of choice mo. So you need to purify it. So kaya, kunyari, sa insulin, kailangan you have to purify the insulin. Yan. Siguro sa factories, kaya mahal to because of the quality control, yung mga equipments na kailangang gawin, okay? So how do you get genes into your bacteria? So make a plasmid with your gene and do bacterial transformation. This is what you'll do in this activity. Yan. So make a plasmid with your gene. So yung plasmid natin na pGLO, binili lang natin yon, okay? Yung scientists in the biorad were the one who engineered those plasmids. Yan. So your plasmids, guys, are circular loops of DNA. Yan. So bacteria can also take in new plasmid. Okay? So more than one kaya yung kaya niyang plasmid. Yan. So perhaps isa lang yung chromosome, pero yan. Your plasmid, you could insert it in your E. coli. So your scientists can modify or engineer the plasmid. Yan. So just an overview. Yan. So meron tayong origin of replication. Yan. Let's the bacteria make copies of the plasmid. Yan. So ang shortcut nito na name niya is ORI. Yan. We also have areas in the plasmid that will produce specific protein or specific trait. And there's also a place in your plasmid which will allow it to have antibiotic resistance. Yan. Hindi ba yan napag-aralan natin last time that yung mga plasmid, they give a genetic information na good add-ons sa mga bacteria. Yan, like antibiotic resistance. So one of the genes that they placed in your pGLO plasmid is a gene that codes for your green fluorescent protein. Okay? So hindi yung protein yung nilagay nila sa plasmid, correct? Yung gene ng GFP ang nilagay nila sa plasmid, okay? So itong mga Aquaria Victoria jellyfish, this is what they look like under visible light, and this is what they look like under ultraviolet light. So parang green yung itsura niya, okay? So since yung genes para sa GFP inilagay natin sa bacteria, what we're going to expect are green neon color, okay? Hindi blue or white. Okay, green neon color under ultraviolet light. Yan. It glows green. Yan. So we will going to be specific on the specific names of the areas of the pGLO plasmid. So padagdag yung mapag-aaralan natin. So itong red one, yan, not necessarily red in vivo, but it's your beta-lactamase gene. Okay? So kung naalala nyo sa atin ba yun? Hindi, sa micro ko yun. So, ang mga beta-lactams, guys, beta-lactams, these are antibiotics that attacks the cell wall of your, or peptoglycan of your bacteria. Okay? Ang beta-lactam drugs. Okay? So, to fight your beta-lactam drugs, example is ampicillin. Yan. Yung bacteria, they could produce an enzyme which is beta-lactamase, anti-beta-lactam drug. So kung may beta-lactamase sila na gene that codes for this enzyme, yan. So ang pinag-usapan natin gene pa lang guys. So if this bacteria has a gene that codes for this enzyme, so it will be resistant to ampicillin, which is an example of a beta-lactam drug. Okay, so kung ang plate may ampicillin, okay, may antibiotic na ampicillin dun sa culture, however, merong pGLO plasmid, your E. coli will still thrive. Okay, mag-grow pa rin siya, okay? Because it has beta-lactamase which could neutralize or make your ampicillin ineffective. Okay? So kahit may ampicillin sa culture carry bells, it's thriving. Okay? So next is your ARO-C. Okay? So, ang ganda ng pagka-explain niya dito na itong ARAC, it's like an on and off gene for your green fluorescent protein. Okay? So, itong RAC, para gumana siya, kailangan niya ng Arabinose. Okay? Sugar. Yan. So, kung present ang Arabinose, yan, pwedeng mag-glow yung GFP. So, may may specific rin ng GFP. So, hindi siya basta-basta mag-glow class kung walang Arabinose. Okay? So, sa culture plate nyo, tingnan nyo mamaya na ang mag-glow lang na culture plate is when it has an Arabinose kasi kailangan nitong RC. Yan. And it has a ampicillin, so sure ka na may pGLO siya kasi it survived ampicillin. It was resistant to ampicillin. Yan. So, para mag-glow, para mag-switch on yung GFP protein, kailangan ng Arabinose. Yan. It's interesting because this is a regulatory protein. So, it regulates your GFP. Yan. Kinokontrol niya yung GFP from activating. Okay? So, ma-activate lang ang GFP, mag-glow lang ang E. coli kung nag-on itong RSE. And the prerequisite, the requirement para siya mag-glow is Arabinose. Okay? Yan. So, I got a picture of, mas mahaba pa, ito ang simple lang, mas mahaba pang mapa ng pGLO. According to Biorad, kung Biorad ang gumawa ng pGLO, it has 5,371 base pair. Yan. So, we have this ORI, itong site of replication or original site of the replication. We have the RC, so the switch that allows your GFP to turn on and off. This gene will code for the green fluorescent protein, and this one, AMP, yan, is the one that BL yun eh. BL is the one that will gene that will cause your beta-lactamase to be produced. Yan. AMPR, wait, I'm not sure what's that. Tingnan natin. Kung ma-search ko. Wala sa ano yan. Anyway, hindi ko mahanap yung AMPR. Anyway, this is where the site, or baka yun na yun, BL, ampicillin resistance, or BL. So also, we have your PBAD promoter. So, pag sinabi nating promoter, guys, this is the sequence na hinahanap, promoter, o kumbaga, promotor. Yan. It is the one that initiates it binds the ROC Arabinose and promotes RNA polymerase binding and transcription of GFP gene. Hindi ba naalala niyo yung may data box, ganyan? So, it was the promoter. So, pag meron na yung data box, lalapit na yung RNA polymerase. And the same thing with this PBAD promoter. So, pag andyan na yung PBAD promoter, tatawagin niya na yung RC, tatawagin niya na yung RNA polymerase, and they will start copying the GFP gene para maging mRNA. Okay, next. We also have multiple cloning sites. So ito, class, mapapansin nyo itong mga perpendicular lines ito. Ayan. So hindi ba mukhang hinahati nila yung pGLO, Plasmid, because these are restriction sites. Yan, na pwedeng gupitin ng mga restriction enzyme. Okay? So if we have ND1 restriction site, this site could be cut by a restriction enzyme also called as NDE1. Okay? Ito, baka mali ako ng basa, CLA1. So kung may CLA1 restriction enzyme ka, pwedeng gupitin to ng CLA1 restriction enzyme. Yan. So, ganoon. So, these are cutting sites, yan, which would be cut by the specific enzyme. So kung NDE1 ang nakalagay dyan, NDE1 lang ang pwedeng gumupit dyan. Okay? So something like that. So guys, just a reminder, your pGLO is made up of DNA, okay? And your DNA has negative charges kasi it will come into play later. Yung role ng calcium chloride, okay? And this is your bacterial membrane. So the challenge, guys, is paano natin maipapasok yung pGLO plasmid sa loob ng plasma membrane. So itong blue, the outer layer of the plasma membrane, is polar. And the inside is non-polar. So, paano kaya natin makapasok yung plasma membrane? Okay, how would the plasmid enter it? So, first step is yun nga. We want to add the plasmid. How? Paano mo siya ipapasok? And so, just a reminder, your plasmid has a negative charge because of the phosphate group or negative charges on the DNA backbone. Yan. So, paano natin mapapasok? Ito na. Dito na pumapasok yung calcium chloride or transformation solution. Yan. So, we're just going over the step, kumbaga, overview or the functions of the different reagents and activities in the pGLO bacterial transformation. Mamaya, bigyan ko kayo ng overview ng steps. So, para pumasok yung plasmid, guys, na may negatively charged, dito pumapasok yung transformation solution, which is calcium chloride. What the calcium chloride is, it covers or shields the negatively charged. So, parang tinatakpan niya, kung bagay yung negatively charged particles to make it less polar. Kasi ang pinapasok niya na yung tong blue part is non-polar. Hindi ba ang mga non-polar glass sa cell membrane mas madali pumasok? Example, mga steroids, yan, mga fatty acids, mga vitamin A, vitamin D, vitamin E, vitamin K, mga addict. They could easily pass through they could diffuse through the cell membrane because they are non-polar substances or they are less polar. So yun ang gagawin ng calcium. It will make it less polar to increase the chances of the plasmid entering the cell membrane. Okay, what else? Aside from transformation solution, we shock it with heat. Yan. So pag heat shock, we expose it to heat, then expose it to a cold temperature. Okay? So if I'm not mistaken, we expose it at 42 degrees Celsius. Yan. Mamaya, tignan natin kung tama ko. 42 degrees Celsius. Painitin muna natin. And what's the function of heating it immediately? Add heat to create pores in the cell membrane para magka-butas. Okay? And what else? The plasmid enters through the cell through the pore by heating it. Okay? And class, immediately we place it on ice after 2 minutes para mag-close ang pores. Okay? So far, what we did is we exposed them to calcium chloride. The calcium will shield the negative charges on your plasmid. Then we heat it at 42 degrees Celsius to open up the pores. Ayan, the pores pala ng cell membrane. And to immediately close the pores, we place it on ice immediately for 2 minutes. Okay, para nasa loob lang yung plasmid. And now, we place our bacterial culture into our broth, LB broth, Loria broth, and allow the gene expression for 10 minutes. So, actually, ang bilis nito 10 minutes lang. So, incubate mo yung bacteria sa broth. Since sa broth, hindi ba may nutrients? So, it's a conducive environment to convert your DNA or your genes into protein. So, wow, ang bilis nito, 10 minutes lang. Yan. So, 10 minutes now, your plasmid, ito lang yung mga main na proteins. It is starting to create beta-lactamase. It is starting to produce RAC proteins from your gene and your GFP. Yan. So, ang GFP, magpro-produce lang siya kung may Arabinose. Okay? So, if the RAC allows it, kung may na-detect siyang Arabinose, doon lang ma-produce tong RAC, at tong RAC, it could start the translation of your GFP. Yan. So, yan. GFP will only be produced if Arabinose is present in the media. So, guys, dito rin yung malalaman the functions of regulatory proteins. So, ang role niya, mag-regulate para hindi mag-overproduce. Yan. So, it's a switch on and off. Okay, same with your proteins, with our body. Okay, hindi rin okay if there is excess accumulation of a certain protein. Okay, example, with your Alzheimer's, yan. Example, may excess production ka of amyloid in the brain, it could produce to Alzheimer's, yan, protein. Tapos hindi na breakdown yung protein na yon. Nako, di ba? Talagang lalala yung dementia mo, yung pagka malilimutin mo. So in the body, if there is excess protein creation from our genes, yan, it could cause disease. Okay, that's the value of regulatory proteins. Yan, galing, no? Yan. So guys, and kunyari, yan, so nalaw mo na overnight, mag-grow, or ma-incubate, tapos nalagay mo na sa selective media, itong, tingnan natin kung, so pumasok na sa loob ng E. coli yung plasmid. So the E. coli has created proteins, yung red, which are your beta-lactamase proteins, di ba? So these are enzymes against your beta-lactam bacteria or your ampicillin. So, notice guys, hindi lahat ng E. coli nagkaroon ng plasmid. Okay? So, ibig sabihin, pag ginrow mo to sa ampicillin, yan, ang mag-grow lang yung may mga pGLO, plasmid, okay? Mamamatay yung mga hindi nakakuha ng pGLO, plasmid, yan. So, that's a way on yan. So yan, nung naglagay ka na ng ampicillin sa plate, yan, they died, yung mga hindi nakakuha ng pGLO plasmid. So this is good. At least, this is a good sign na kung i-isolate mo yung plasmid, at least yung mga bacteria na makukuha mo, sure lahat sila may plasmid. Okay? That's our next topic. So this is a good way on removing anything which doesn't have the plasmid. Yan, using a selective media. So the transformed bacteria with the plasmid will make beta-lactamase, yan, which breaks down ampicillin. This enables them to grow on ampicillin plates. Bacteria without the plasmid, if they are not transformed, could not grow on plates with ampicillin. Naintindihan ba, guys? So ito, they are thriving in the media with ampicillin. Yung mga walang plasmid, they're dead, okay? They're history. So, ang ginamit nating broth is Luria broth or lysogeny broth, okay? So, this contains broth talaga. Ayan. It's like a chicken noodle soup for bacteria. Hindi ba mga chicken noodle soup? It strengthens you. Di ba pag may sakit ka, ang sarap uminom ng chicken noodle soup. It has all the nutrients your bacteria need to grow. Eh, di rarami sila. Di ba? Pag dito mo sila ginrow. Okay. So this, guys, are the summary of the reagents. So yung calcium chloride, it will shield the negatively charged on the DNA, allows the plasmid to enter the membrane. Yan. So it increases the chances of allowing the plasmid to enter the membrane. Yan. Pre-heat shock incubate, ito muna. Heat shock muna. So based sa slow fluid plasma membrane for greater shock. Okay. Okay. So, palamigin mo muna. Tapos, painitin mo. Tapos, palamigin mo ulit. Okay. So, palamigin mo muna. Slows the fluid plasma membrane for greater shock. Then, heat it to increase to allow the opening of the pores, increase the permeability, then place it on ice. Yan, to restore the cell membrane, to return it back, to close the pores that you open. Okay, so heat it, preheat shock incubation on ice. Sorry, ice, then shock it with heat, tapos ice na naman. Okay. And incubation at room temperature with L'Oreal broth. Yan. So, 10 minutes to allow gene expression. So kumbaga, bigyan mo naman ng time para mag-produce sila ng protein. So, allows the expression of beta-lactamase. Yan. Para mag-survive siya sa ampicillin. So, if you did not incubate it, so wala pang time gumawa ng beta-lactamase, edi mamamatay sila. Okay? So, if you miss this, yan, mamamatay yung mga E. coli mo sa ampicillin plate. Okay, please remember that. So, don't forget to incubate it at L'Oreal broth for 10 minutes. And spread it on your L'Oreal broth / ampicillin plates. Selects for transformed bacteria and allows formation of colonies. Okay. So, lahat ng mga andito, yung makukuha mo dito is lahat sila may pGLO, Plasmid. Sure ka nun? Okay. So, ito na yung mga step-by-step procedure sa pGLO natin. Yan, balikan lang natin. So, meron tayong microcentrifuge tube number 1 and microcentrifuge tube number 2. So, first tube, yan, lagyan mo ng okay, for the two tubes, add 250 microliter transformation solution or calcium chloride. Okay? Tapos label it, positive pGLO. So meaning, lalagyan mo ng pGLO, plasmid, itong tube na to, and negative pGLO, walang pGLO, plasmid. Okay? So, ibig sabihin, this is our control. Okay? So, syempre, since hindi ka naglagay ng plasmid dito, okay, dapat walang tutubo sa ampicillin. Pag ginuro mo itong negative pGLO sa ampicillin, dapat walang tutubo. It should be dead because they are not resistant to ampicillin. Okay, they are sensitive to ampicillin kasi nga hindi mo siya nilagyan ng pGLO eh. Okay, so just label it first and place it on ice. Okay, so calcium chloride, yan, usually kailangan naka-ice yan. Then, add your colonies of E. coli, yan, on both tubes. Then, place it on ice, yan. So, you could use a vortex para to homogenize it, yan. Using new loop, so lagyan mo ng E. coli yung dalawa, okay? So, dapat brand new loop yung dalawa so that you could not contaminate. So, pag itong pGLO, nilagay mo yung inoculation tube mo, kumakain naman ng E. coli, nilagay mo dito, dinalagyan na ng pGLO yung negative mo, di ba? So, always use a new inoculation loop, then place it on ice. Place it on ice. Then, add 10 microliters lang of pGLO on the tube where it says positive pGLO, okay? At wag mong lalagyan yung negative pGLO and place it on ice for 10 minutes. Yan, parang lagi lang nasa ice yung microcentrifuge tube. So, the only time we are going to place it is sa heat shock. So, while doing that, yan, incubate mo muna. So, prepare your plates. Yan. So, dito mo ilalagay yung pGLO na lagyan mo lang ng label lang. Negative pGLO, L'Oreal broth. Itong negative pGLO, L'Oreal broth, and ampicillin. Yan. Siyempre, dapat walang growth dyan. Dito, sa pGLO with ampicillin, dapat may growth dyan because it's resistant to ampicillin already. And ito dapat, since may Arabinose to, kailangan, by the end of the experiment, dapat nag-glow to kasi may Arabinose, di ba? So, your Arabinose is required so that your pGLO will produce green fluorescent protein, okay? So, hindi Arabinose ang the presence of Arabinose will activate your ARA-C gene, okay? And this ARA-C gene will allow the transcription and translation of pGLO, okay? So, yung Arabinose mahalaga, yung ARA-C gene mahalaga para ma-produce yung pGLO. Okay, so get your timer ready. So heat shock your tube at 42 degrees for exactly 50 seconds. Yan, to allow the pores to be open. Yan, and immediately return the tubes in ice for 2 minutes. Yan. Then after doing heat shock treatment, yan, so you could now add L'Oreal broth to both of your pGLO and negative pGLO tubes. So, add 10 minutes. Yan. So, 10 minutes to allow the expression of your beta-lactamase. Okay. So, meanwhile, what's happening inside? Your E. coli, nakapasok na yung plasmid kasi in-expose mo naman na siya sa calcium chloride, saka sa heat shock. And it will now be expressing genes, the genes are expressed through the formation of proteins. Okay. And so the red one, yan, beta-lactamase protein. So enzyme class are protein. Okay. So yun na rin. So your RAC is a regulatory protein. Yan. So once it detects Arabinose, it will turn on the transcription and translation of GFP. Kakalimutan. Yan. RAC controls the expression of GFP. Kakalimutan yun, ha? So Arabinose sugar works like a switch. Without Arabinose, the switch is off. It blocks RNA polymerase. The RAC blocks the RNA polymerase and the GFP gene is not transcribed. Ooh, tingnan natin ulit yung picture. Yan, RAC will block. Yan, block wow, galing. Oh, sapa. Yan, it will block the site. Hindi makapasok yung RNA polymerase, hindi siya makakagawa ng mRNA out of the GFP. Yan. So, it works like a switch. Yan. So, galing. So, if the Arabinose is wala, the switch is off, RAC will block the RNA polymerase ulit. Yan. And with Arabinose, the switch is on. RAC changes shape and RNA could transcribe the GFP. Okay, tingnan nga natin yung shape niya kanina. So, nung hinarangan niya. Yan. Wait lang, break tayo. Yan, dito, hinarangan niya. Okay. Kasi walang Arabinose. Next, there is an Arabinose. It changes shape and allows your RNA polymerase to perform transcription. So, para makagawa na ng mRNA out of your GFP gene, tapos tong mRNA, babiyahin na sa ribosome, tapos magproproduce na ng green fluorescent protein. Okay? So, ang RAC is a regulatory protein. Yan. So, if Arabinose is present, it will not block your RNA polymerase. Okay. So, yan, since the Arabinose is present, GFP is produced. Okay. Yan ang magproproduce ng green fluorescent light. So now, after the incubation, yan, nakaproduce na ng beta-lactamase. Flick the tubes to mix, yan, or you could use a vortex. Using a new sterile pipette, add 10 microliters bacteria to your plates, yan. So ito yung positive pGLO. So lagyan mo yung positive pGLO, LB, and ampicillin positive pGLO with the LB, the ampicillin, and the Arabinose. And dito, 100 microliter, yung negative pGLO dito sa negative pGLO, LB/ampicillin, and LB lang. Okay, so madali lang naman guys, diba? Incubate overnight at 37 degrees or 2 days at room temperature. Okay. At ito yung summary of the contents of the plates. Yan. Siyempre, yung buong plate, nung nilagay mo na, lahat sila may presence of bacteria. Lahat sila may E. coli, okay? So, dapat, meron at meron mag-grow, okay? Kasi halos lahat sila naman may E. coli, except itong may ampicillin at saka negative pGLO. So, walang growth talaga. Kasi, since hindi siya naka-receive ng plasmid, and E. coli at normal state, it's not resistant to beta-lactams or ampicillin, dapat walang growth talaga. Okay? So, itong DNA, so, negative dito, positive dito, kasi nga, yun lang yung positive pGLO. Okay? They receive DNA or plasmid. Alin yung may ampicillin? Tatlong plates. Okay? So, ito, tsaka ito. Arabinose, syempre, yung dulo lang ang may Arabinose. Okay? So, expect yung tube na may Arabinose, may plasmid pa, yan, expect there should be a glow. Okay? Siya lang mag-glow. And for the growth, guys, yan, since yung pGLO, resistant naman siya sa ampicillin, it will grow. Okay? Tapos, ito, mag-grow rin siya. So, hindi siya mag-grow kasi wala siyang plasmid, and it's sensitive already to ampicillin, and ito, mag-grow siya, okay? Kasi nga, Loriobrose lang naman ang laman, okay? Pero, ang mag-glow, isa lang, yung may Arabinose. Magandaan ba, guys? So, negative pGLO, there is a confluent growth. Dito, negative pGLO with ampicillin, walang growth. May growth naman dito sa positive pGLO. Pero dito sa positive pGLO with Arabinose, ang mag-glow. Okay. So, notice, yan. So, walang growth dito. Yan, may growth dito, hindi umilaw. May growth naman dito, pero hindi umilaw. Pero dito, may ilaw kasi the presence of Arabinose. Yan. So, guys, that's it for today. Okay, and finally, we have transformation efficiency. So, how efficient was the transformation? Kasi nga, I told you hindi lahat ng E. coli ay natatransform. Okay? Some E. coli is unable to be transformed. So, how could you calculate the transformation efficiency? The total number of colonies growing on the agar plate and the amount of DNA spread on the agar plates. Yan. So, if there are 87 colonies growing on your plate, over 16 micrograms of DNA spread, I think, yan, hindi ko alam kung paano mini-measure to actually. So, anyway, example lang, equals 543 transformants per microgram. Yan. So, I was searching for a reference value. Wala akong mahanap. So, guys, yan na ang discussion natin today. I wanna thank you. Bye-bye.